So welcome back again from the break, whether you stepped away for a moment or whether you just clicked through to continue on. Let's take a look at the concept check question, and then we'll move on to the eukaryotic regulation of genes. So mutation in the E. coli makes the lac operator unable to bind the active repressor. So the repressor is not going to bind to the operator at all. How would this mutation affect the cell? Well, in the absence of a bound operator, remember, that means that the operon is turned on because it's the binding of the repressor to the operator that keeps the gene or the operon switched off. So if you've got a mutation that prevents the repressor from binding to the operator, then that means that the operon is going to be permanently on. So that's how it would affect the cell. Would this be a disadvantage? Absolutely, because now the cell is investing resources in making enzymes and other proteins that are not necessary because there's no lactose present. Remember, the operon is only supposed to be turned on if lactose is present. But in this circumstance, where the operator is not binding to, or the repressor is not binding to the operator, that's going to leave the operon permanently switched on and mean that it's going to be investing resources and energy in making something that it does not need at the time. So that would be a distinct disadvantage to the cell. So when we start thinking in terms of eukaryotic cells, the regulation of genes here is way more complicated. And we, in fact, draw a little flow diagram. The book has drawn it as a series of pipe arrangements, done it as a plumbing analogy, with a series of valves along the pipes, and each one of those valves represents a regulation point. So if we take a look at that diagram, here you can see what it looks like. So we have the original chromosome, and the chromosome, as you may recall, is packaged within the nucleus so that it can actually fit inside the nucleus. And that packaging, in part, helps regulate the expression of genes, as we'll see. In order to be able to express the gene, the area of DNA in the chromosome that represents the genes actually needs to be unpacked so that it's accessible to the RNA polymerase for transcription. So there is a regulation point here in terms of the packaging or unpackaging of the DNA. Then we have the actual process of transcription. And if you recall from our discussion of transcription in eukaryotes, there are several ways in which that the gene can be regulated, most notably in terms of the presence of introns and exons in the transcript and the requirement to cut out the introns and splice the exons together. We're going to talk about that in more detail. I gave you a hint of it when we talked about transcription, but we're going to talk about that in a little bit more detail as we work through this flow diagram. So there is a means of regulating gene expression in terms of cutting out the introns and splicing exons together. Then we also need to protect the messenger RNA once we've cut out the introns and spliced the exons together, and that involves putting a cap on one end, on the 5' end, and putting what is referred to as a poly-A tail on the other end, the 3' end. And that the length of that poly-A tail determines how long the messenger RNA is going to remain stable in the cytoplasm and be available for the ribosomes to translate. So that's another point at which we can regulate the expression of the genes. So the lifetime of the messenger RNA is another regulation point. Once we start getting into translation, then we can regulate the polypeptide itself in terms of whether it is translated or not and what polypeptide is made. And then once we've got the polypeptide made, most polypeptides need some form of modification. and we could talk about insulin, or we could talk about a number of other polypeptides there that need to undergo various changes in order to become active. And then finally, we can talk about the actual lifetime of the polypeptide or the protein itself, because proteins do not have an infinite lifetime. They eventually get broken down. And there are ways for cells to identify which proteins need to be broken down and when, by using tags, and we'll talk a little bit about that when we get to it. So we've got another checkpoint question here, and I specifically didn't take this one out because I want us to take a look at this for a moment. So of all of the control points along here in this flow diagram, which is the one that you would consider to be under the tightest regulation? So obviously I can't hear what your answers are, but if you had in mind that it was the packaging of the DNA, then you're correct. Because that's where the tightest control is, because if the DNA simply does not get unpacked at all, then the genes that are in there never get transcribed. And so when we think in terms of gene expression giving rise to different types of cells, one of the things that we immediately recognize is that one way for the cells to determine which proteins to produce and which proteins not to produce in order to become the types of cells that they are destined to become is simply to leave those areas of the chromosomes that contain genes that they don't need to express fully packaged and inaccessible to the RNA polymerase. And then where there are genes that need to be expressed, have those unpackaged so that they are available for transcription. It is a way of regulating what a cell will become. And we'll talk a little bit about how they get the signals to do that sort of thing a little bit later in this presentation as well. This presentation is actually full of a lot of information. It's quite an extensive presentation. So this is the area. Just make sure the pen is on. So this is the area where the tightest regulation is taking place, in the packaging of the DNA. Because if the DNA is simply not unpackaged, it can't be accessed by the RNA polymerase, it can't be transcribed, which means there's no messenger RNA, which means there can be no translation, which means there can be no protein. So long as you keep the DNA packaged and inaccessible to the RNA polymerase, and prevent transcription, you permanently keep the gene turned off. And that is the situation in a lot of cells. The vast majority of genes are permanently turned off because they are not essential to the function of the cell. A very extreme version of this is something that takes place in all female mammals. You should recall that a female mammal has 2 copies of the X chromosome. And the problem with that is that having 2 active X chromosomes leads to what is known as a gene dosage problem. Too many active versions of the genes on the X chromosomes. What a female mammal actually needs is only 1 active X chromosome. So very early on in the development of a female mammal fetus, and this includes humans, 1 of the 2 X chromosomes in every cell undergoes an extreme condensation event, and it creates a structure called a bar body. This is a very, very tightly packaged X chromosome that is so tightly packaged that it never unpackages again, and that X chromosome is permanently inactivated. It usually takes place at the 8-cell to 16-cell stage in the development of the fetus or of the embryo. And why this is important is because all the daughter cells of each one of those 8 to 16 cells is going to inherit the same X chromosome in an inactive form. And that which of the 2 X chromosomes is inactivated is random amongst those cells. So each cell will randomly have 1 of the 2 X chromosomes, but each cell may have a different X chromosome inactivated compared to its neighbors. It really depends. But we do know that the daughter cells of each of those cells are going to inherit the same X chromosomes inactivated, which means when you take a look at a female mammal, it's possible that different groups of cells scattered throughout the body of that individual are expressing different genes according to which X chromosome was inactivated. Now, in humans, it's very difficult to pinpoint a circumstance where this is the case. But there is a very, very clear example when it comes to thinking about cats. Because the tortoiseshell color in cats is a product of individuals who are not homozygous but heterozygous for a fur color gene located on the X chromosome. Now, if the cat was in fact homozygous, it wouldn't matter which of the two X chromosomes was shut down, because they both contain the same allele of the coat color gene, and therefore the cat would either be orange and a tabby cat if it's the orange allele that's present on both chromosomes, or black if it was the black allele on both of the X chromosomes. But if the individual is heterozygous, they're going to have the orange allele on one X chromosome and the black allele on the other X chromosome. And remember, when one of those X chromosomes gets inactivated, it's a random event. And the daughter cells simply inherit the inactivated X chromosome, which means that when you look at a tortoiseshell cat, what you're seeing is that random distribution of inactivated X chromosomes, because the different parts of the body are derived from different cells within that little ball of cells when the X chromosomes were inactivated. And what is interesting is that tortoiseshell in cats so it's about 96% tortoiseshell cats. Sorry, this is not terribly neat in terms of writing, but hopefully you can read that. So about 96% of all tortoiseshell cats are female, which leaves about 4% of them being male. So if you see a male tortoiseshell cat, so if you happen to come across a male tortoiseshell cat, you can be very confident that his sex chromosome combination is XXY. And this is what leads him to be tortoiseshell, because one of those two X chromosomes gets shut down just as in the female cats. And if he's heterozygous across those two X chromosomes for this coat color gene, then he will be himself tortoiseshell, just like his female compatriot. So when you see a male tortoiseshell cat, you know what its sex chromosome complement is. It's XXY. That's certain in these circumstances. So this is again a form of gene regulation, right? You're changing, or you're changing the expression of the genes by inactivating the X chromosome. It's a very extreme example of what goes on all the chromosomes, because in this case, the X chromosome not only gets inactivated, but it is permanently inactivated. Because having two active X chromosomes would lead to having too many active versions of, or too many active genes that are associated with the X chromosome. So one of the X chromosomes always gets shut down as a way of regulating the expression of the genes on the X chromosome. And we see the outcome of that in the tortoiseshell coat color. As I said, the same sort of thing does go on in humans, but we don't know of any particular conditions that we can pinpoint in humans that are the direct product of this shutdown, this random shutdown of one of the two X chromosomes early on in the development of human females. That doesn't mean that there aren't any. It's just that it's very difficult for us to figure out which ones might be associated with the X chromosome, because the chromosomal system is very different between cats and humans. No surprise there. Anyway, moving along. When we start thinking in terms of how transcription is regulated in a eukaryote, it turns out that the initiation of transcription, once the DNA is unpackaged and made accessible for the RNA polymerase, the way in which you activate the gene, meaning you signal to the RNA polymerase the location of the gene and its promoter region to which it is going to bind, is a little bit more complicated than the situation in bacteria. Now, in bacteria, we know that transcription factors are involved, but the system of transcription factors in bacteria is a lot simpler than the system of transcription factors found in eukaryotes. Now, I'm going to jump over a couple of slides to get to a diagram, and I'll explain exactly what's going on in that diagram. So actually, I didn't need to jump over it, just right here. So you can see the basic makeup of the gene. There is the promoter upstream of the gene, and if you've forgotten what upstream means, you can go back and take a look at the previous presentation, because I defined upstream and downstream there when we talked about transcription. So the promoter region is upstream of the gene, and that's the area that the RNA polymerase is going to bind to. But the RNA polymerase won't bind to the promoter just randomly. It needs to be signaled that it needs to bind to that promoter. So what we now know is the situation is that elsewhere in the DNA, outside of the gene coding region, and in some cases quite a long distance away from the gene, there are short DNA sequences known as enhancers, and you can see them here. There's a pair of enhancers here, and transcription factors known as activators bind to those enhancer regions. That causes the DNA to bend around and bring these enhancer regions adjacent to the promoter region of the gene. And there's actually another protein, you can see it here, DNA bending protein, that actually assists in bending the DNA around and holding the DNA in its bent conformation to bring these activator proteins adjacent to the promoter region. These in turn bind to transcription factors that are going to bind to the promoter. And it's this complex of proteins that attracts the attention of the RNA polymerase so that it will bind to the promoter and start transcribing the gene in this direction. So the activator proteins are what binding to the enhancer regions of the DNA. The subsequent bending of the DNA to bring the activators close to the promoter is what sets up the complex of transcription factors that then aid the binding of the RNA polymerase to the gene so it will be transcribed. So it's a pretty complex setup, but it works. And it allows for eukaryotes to have a very fine control over the activation of genes, because every gene responds to a particular set of transcription factors. And as you'll see, there are proteins that function as transcription factors that are important in determining what genes are going to be expressed and what genes are not going to be expressed in every type of cell. It's what allows eukaryotes like ourselves, and I'll give you some numbers here. We know that in terms of actual genes in our DNA, we contain only around 23,000 genes. Somewhere between 21 and 23,000 genes is the number that most people accept these days. Why that number should be so important is because in terms of the number of genes to make a little Drosophila, the little fruit fly that we've talked about previously, requires 17,000 genes. So in terms of absolute gene numbers, there's not a whole lot of difference between a Drosophila, a little fruit fly, and ourselves in terms of actual genes. But where we have the advantage over Drosophila is the way we utilize those genes. We have far more transcription factors and control factors that regulate the expression of the genes versus what Drosophila have. And because we have this greater level of regulation, we can do so much more with the genes that we possess. We can create a far more complicated body, ours, from only a few more genes than a Drosophila, simply because there's so much more that we can do with the genes that we possess, because we have much more control over them. And we'll talk a little bit more about that control as we go through this chapter. So this animation walks you through the activation of a eukaryotic gene via the transcription factors and the activator proteins. Activation of a eukaryotic gene. And of course, as always, these animations are located on the resource pages associated with this chapter. So this is chapter 11. So you'll want to go to the chapter 11 resource page and you will find this animation, and all the animations are listed in sequence according to how they appear in the presentation. So you shouldn't have too much trouble finding this one. So this animation simply walks you through what I just told you. These are the activator regions down here, or the enhancer sequences that the activators bind to, and you'll see the DNA bend around. The activators will then bind to this transcription factor that binds to the promoter. These other transcription factors then bind to the promoter, which encourages the RNA polymerase, which is this protein, to bind to the promoter region and start transcription. And that's all played out in this animation for you. We also know that there are repressor proteins known as silencers that can also bind to the DNA sequences adjacent to the gene and prevent the RNA polymerase from binding and prevent the complex of transcription factors from forming up. However, there are not as many repressor proteins in eukaryotes as we would expect to find in prokaryotes. Typically, it's activators that are responsible. It seems like the vast majority of genes in eukaryotes are, unless they're required to be active, in a default inactive form. So there's no real need for a lot of repressor proteins to keep genes switched off. What is required is activator proteins to turn the genes on that would otherwise be turned off. And of course, as it mentions here, there are going to be some housekeeping genes, and I've mentioned this before, like the genes involved in cell respiration and so forth, that are going to be kept on in most cells because they're necessary for the general maintenance of the cell. It's only those proteins that need to be turned on at specific points in the cell cycle. Remember, we talked about the cell cycle when we talked about mitosis. So there are various points in the cell cycle where we expect to have proteins present to allow the cell to continue in the cycle. Those are the sorts of things that are going to be turned on and off, as are going to be those genes that are required for establishing what type of cell the cell eventually develops into and what types of cells that will continue to be produced as a product of mitosis when those cells divide. The same set of genes need to be active at all times to maintain the identity of the cell. So it is predominantly activators activator proteins binding to the enhancer sequences that in turn activate the genes by attracting the transcription factors that make it possible for the RNA polymerases to bind to the promoter. So once we've got transcription underway and we finally get a transcript, remember that transcript is not the final messenger RNA. It is just a copy of the gene, or the gene region. But we know from our previous discussion of transcription that eukaryotic genes are laid out a little bit differently from prokaryotic genes. Unlike prokaryotic genes, which are continuous with no interruptions, in eukaryotic genes, there are non-coding interruptions. We refer to those as introns. And so we need to remove the introns. That's one thing that needs to take place. We then need to take the remaining pieces of the transcript, which are the exons, the coding regions. We need to splice those together to create the messenger RNA. But we're not done yet, because then we need to add some protection to the messenger RNA, because if it was sent out into the cytoplasm at this point, it would be immediately digested and there'd be no translation and no polypeptide. So we need to add a cap, which is a modified G nucleotide, to the 5' end, which is the beginning end of the messenger RNA, where translation will begin. And then we need to add at the other end, the 3' end of the messenger RNA, a long repeating sequence of A nucleotides, which could be anywhere up to 150 A nucleotides long. There is a region inside the gene sequence itself, a little bit downstream from the stop codon of the gene, that actually contains a sequence of nucleotides that tells the cell how long to make this, what is referred to as a poly-A tail. Poly-A because it's many, 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 many A's in sequence, as I said, up to 150. And we'll encounter that as we go through the presentation. And you'll see it in mentioned in some of the animations that are included in here as well. So we've got 3 different things that take place after we have the transcript made by the RNA polymerase. We've got to cap and tail it, give it some protection. We've got to cut out the introns, because they're all the non-coding interruptions. And then we've got to splice the exons together. So let's take a look at what happens here. So I've jumped over a text slide to get to this slide, because this slide shows you exactly what happens in sequence as we change through the slides. So here you can see a eukaryotic gene, just an example of an artist's rendition of the typical eukaryotic gene. You can see the exons in here, and then you can see the introns as the interrupting regions, the light blue stages. So the first thing you're going to do is make a transcript. And the RNA polymerase does not discriminate between the exon sequences and the intron sequences, which is the reason why we need to process the messenger RNA, the transcript, because the RNA polymerase can't differentiate between the introns and the exons. It just copies everything. So once we've got the transcript, then we need to cut out the introns. So we cut out the introns, and then we need to splice together the exons. But here's where it gets interesting, because there is no rule that says, number one, we have to use all the exons and splice them together to make the final messenger RNA, nor is there any rule that says they have to be spliced together in a particular order. So here you can see 2 resulting messenger RNAs that have been produced by splicing together exons, yes, but not necessarily using all the exons. So in this case, we've got exon 1, 2, 3, left out 4, and put in 5. And that gives us a messenger RNA. But if we take the same exons and instead use 1, 2, leave out 3, and put in 4 and 5, we've got a different messenger RNA that's going to produce a different polypeptide. And we know that this is what takes place in eukaryotes. We know that there are systems that depend on being able to splice together exons in different orders and different combinations, the most notable of which is your immune system, specifically that part of your immune system that makes antibodies, because there are a limited number of genes that are involved in making those antibodies, and we have a pretty good idea of how many genes are involved. But the immune system is able to make antibodies against a significantly larger number, by several factors of 10, larger number of pathogens than the number of genes that are involved in making antibodies. And it puzzled everybody for a long time as to how the immune system could make all of these antibodies using such a small number of genes, comparatively speaking. The answer is, of course, the ability to be able to splice together exons in a number of different combinations, leading to a number of different possible antibodies being produced from a limited number of genes. And once this was recognized, several different research groups sat down and ran the math of how many possible antibodies could be produced by the limited number of genes, taking into account the number of possible ways you could combine the exons together to create different components of the antibodies. Specifically, the component that's in question here is what is known as the variable region of the antibody. So there are two regions in an antibody. There's a fixed region that is recognized by the cells of the immune system, and then there's the variable region that actually binds to the pathogen, and that's the one that needs to change from antibody type to antibody type to be able to recognize different types of pathogens. So not surprisingly, it's the genes that produce the variable regions of the antibodies that have these introns and exons and are able to splice the exons together in a number of different ways to produce different types of variable regions for antibodies. And when these research groups ran the math, it turned out that there was a nice correlation between the number of different types of antibodies you could produce and the number of pathogens that the immune system could actually recognize. In fact, there was an extra capacity built into the system to be able to recognize new pathogens as time went by, all because of this ability to splice together exons in different ways, in different combinations, to produce different polypeptides from the same gene. This has obviously caused us to reflect a little bit more about what is the nature of a gene and what does a gene produce. And we used to think that one gene produced a single polypeptide. That's not the case anymore. We can't really say that, because here is an example of one gene with the capacity to produce more than one type of polypeptide on the basis of how the exons are spliced together. But again, this is another way of regulating gene expression, because if you go in this direction, you get a polypeptide. But if you go in this direction, you get a different polypeptide. And so by splicing the exons together, you are actually regulating the expression of the gene by regulating which of these two polypeptides are being produced from this gene by changing the way in which the exons are spliced together. It is a form of gene regulation. So this animation shows you the variation in exon splicing. So that's what this animation is all about. So you can take a look at that one. And then this animation looks at the longevity of the mRNA as a result of polypeptide poly-A tail. So this animation just reinforces how the length of the poly-A tail affects the longevity of the messenger RNA based on what we know about the size of poly-A tails and how that correlates with what we've seen in terms of the longevity of messenger RNAs. So again, by regulating how long the messenger RNA is active in the cytoplasm, you are again regulating the gene expression, because if the messenger RNA is around for a long time, some messenger RNAs can be present for weeks, then it's available for translation during all of that time, and you can make a lot of protein from that messenger RNA over the course of time that messenger RNA is around. If, however, the messenger RNA is only present for an hour or so, you have a limited amount of time in which to make protein before it's degraded. So there is a limit on how much protein is being produced from that messenger RNA. Again, it's a way of regulating gene expression by regulating how much protein is being produced by regulating how long the messenger RNA is available for translation. If it's available for translation for a long period of time, a lot of protein. If it's present for a short period of time, not so much protein. It is again a way of regulating gene expression by regulating the lifespan of the messenger RNA. Then another way of regulating gene expression at the translation level is via some small RNA molecules, and there are two in particular that are of interest here. There's a type of RNA molecule called a microRNA molecule. Like all RNA molecules, it's single-stranded. And then there's another type of RNA molecule called a small interfering RNA, or an siRNA. Now, we know a little bit more about the microRNAs than we know about the siRNAs, but I'll tell you about both of them because they're both relevant. So this diagram illustrates what happens with the miRNAs, or the microRNAs. If the microRNAs are fully complementary to a target messenger RNA, in other words, the sequence of nucleotides in the miRNA exactly matches the nucleotide sequence in the messenger RNA, the whole length of the miRNA is going to bind to the messenger RNA. Now, it does so in concert with a protein that it complexes with. And the presence of that miRNA attached to the messenger RNA triggers the protein to then break the messenger RNA down. In other words, it's a way of removing the messenger RNA. So long as the miRNA is fully complementary to the sequence of nucleotides in the messenger RNA, then the messenger RNA is going to be degraded. If, however, it is only partially complementary to the messenger RNA, it won't trigger the breakdown of the messenger RNA, but it will block the ability of the ribosome to translate that messenger RNA. So that effectively shuts down translation anyway. So one way or another, the miRNAs prevent translation of the messenger RNA, either by being completely complementary to the sequence of the messenger RNA and leading to its breakdown, or being partially complementary to the messenger RNA and consequently acting as a block to the ribosome so they can't translate the RNA. Either way, you're not going to get a polypeptide being produced. And again, it's a way of regulating the expression of the gene. With the siRNAs, I don't have a diagram for that one, but I can walk you through that. The siRNAs bind to the area of the messenger RNA that the ribosomes would attach to initiate translation. And by binding to the region of the messenger RNA that the ribosomes are going to form around in order to initiate translation, you simply block translation. And we know that this is a way of regulating translation because we actually use this to our own purposes. Because we can make synthetic siRNAs and inject those into cells to block the expression of particular genes so that we can find out what that gene does in a cell. Now, I don't have the slides to show you this in this presentation, but for one of the presentations that I do for the General Biology 2 class, where we talk about embryological development, one of the key processes that needs to take place during the development of an embryo is for cells to attach to each other in the right combinations. And that is done by a group of proteins called cadherins. These are proteins that allow cells to attach to each other in particular combinations to form structures. And one of the experiments that we talk about in that presentation is the blocking of a particular cadherin right from the very beginning of embryological development. In fact, even prior to fertilization, by simply injecting an siRNA into the egg cell. It is maintained inside the egg cell, and when the messenger RNA for the particular cadherin protein that is targeted in this experiment finally appears in the cytoplasm of the cells that are derived from the zygote, they block the translation of that protein, which leads to the complete disorganization of the developing embryo, or because of the absence of that one protein. That's how significant these RNA molecules actually are. It probably is going to turn out, and this seems to be a general trend, that the RNA molecules and their ability to regulate gene expression at a number of different levels, remember we've seen them operating at a number of different levels here, may turn out to be way more important than thinking about the role of proteins in regulating gene expression. It may very well be that these little RNA molecules, and we're only talking about a few of them right now, these little RNA molecules may be more important in regulating gene expression than we might have imagined, because we are seeing them turn up in a number of different locations, doing a number of different functions that are all involved in expression of genes, or blocking of gene expression, or transporting proteins to particular locations, or degrading proteins in some situations. So these little RNA molecules may actually turn out to be very, very important cell regulators. That seems to be the situation. So once we get through the process of translation of the messenger RNA and we've finally got our polypeptide, then there's another way of regulating the expression of the genes, by regulating the activity of the polypeptide. So it turns out that many polypeptides need to be modified in some fashion. This typically goes on in the Golgi apparatus, where the proteins will be temporarily stored before they're transported on to their final destination. In the case of insulin, it needs to actually be cut into two pieces that are held together by cross-links that are known as disulfide bridges. So you have two pieces of polypeptide that are linked together by these cross-links, and that represents the active hormone, or the active insulin hormone, that is exported from the pancreas into the bloodstream to regulate blood sugar. And in fact, that's what this animation illustrates. So here you can see the what is called pro-insulin. And if you follow through this animation, it shows you how the insulin is activated. It also shows you right at the very end another way of regulating a polypeptide as well, by adding different types of molecules. Most commonly, it's through adding sugar residues, which creates a molecule that we refer to as a glycoprotein. And you may have heard that term before, but it's the typical addition to a polypeptide. If we're going to add something to a polypeptide, is the addition of sugars, and that gives us a glycoprotein. Another thing that you might do, of course, is add energy to it. We've talked about activating proteins by adding energy to it. So that would mean the transfer of a phosphate group from an ATP. All of that is also illustrated here in this animation. So this animation actually talks about how to activate polypeptides. Cutting the polypeptide in the case of insulin, addition of sugar residues, creating glycoproteins, or addition of energy via the transfer of a phosphate group from ATP. These are all ways of activating proteins, activating polypeptides. So if those steps don't take place, obviously the proteins don't get activated. So again, you're regulating the expression of the gene by regulating the activity of the protein, because the ultimate expression of the gene is the active protein. If the protein is not activated, then you really don't have the gene being fully expressed. You just have a polypeptide that's doing nothing. That's not very helpful to the cell or beneficial to the cell. Then this animation shows you how polypeptides can be deactivated. In other words, cut up. And this is activated by the addition of a little protein called ubiquitin. Why is it called ubiquitin? Because it is ubiquitous in the cell. There's a lot of it in the cell. And so when ubiquitin molecules are added to a protein, that signals to other complexes of protein that this protein, this polypeptide, needs to be degraded. And so it is degraded by these proteases. They are enzymes that break down proteins, and recycle the resulting amino acids are then recycled back into new polypeptides and proteins. So when a protein has served its function, when it is no longer necessary, it gets tagged with ubiquitin. That signals that it should now be broken down. These big complexes come in, break them down, and recycle the amino acids back to be used for generating new polypeptides. Again, this is a way of regulating gene expression because it determines how long the protein is active. Remember, again, the ultimate expression of the gene is its active protein. So if you allow the protein to hang around for a while, you have extended the expression of the gene because the protein is being active. If you break down the protein, if you destroy the protein, then you have influenced the expression of the gene by limiting the amount of time that its protein is active. So let's take a break for a moment, and this will be a full break. So I've got a concept check question, which I will answer when we come back on the other side of the break. Name 3 ways that the RNA may be processed after a gene is transcribed in the nucleus. Something to think about. I'll see you on the other side of the break with the answer.